Hello everyone, you're welcome to biology video lectures by Akerele Oladimeji Philip. How has been your week and all? On today's edition of the class, we'll be looking at the cell. Alright, and this will be divided into about 4 segments. So this first segment deals with cell and its structures. Alright, so cell is defined as the basic structural and functional unit of life. That is what the cell is. That means the cell, what that statement means is that the cell has the ability to carry out Mr. Niger Dark, talking about the acronym for characteristics of living things. So for a cell to be called the basic structural and functional unit of life, it means that it can carry out all life processes, which is majorly embedded in those characteristics of living things: movement, reproduction, nutrition, irritability, until you get to the last acronym, that letter on the acronym D, which is death. Alright, now there are actually theories that backs up cell's existence. Yes, you know, in science, theories, laws are made so that it is ascertained that what is being talked about is valid in all respects. So there are actually about 5 theories. This theory is not meant to be listed in order. It can be listed anyhow. It doesn't mean the fact that I wrote this as the first one has to be the first one at any time, any day. No. So it could be anyhow. Alright. So now there is no life apart from the life of a cell. That means that everything an organ or an organism can do starts from a cell. Let me explain what that means to you in a minute. Now, cells form tissues, tissues forms organs form system, and systems forms the organism. Now, if we say that a cell can carry out characteristics of living things, basically, which is true, that means that if we kill the cell, we have killed the tissues. If we've killed the tissues, we've killed the organs. And if we kill the organs, we've killed what? Systems. Killing the systems, you have killed the entire organism. So that's what we say. There is no life apart from the life of the cell. So everything, even as human, every of our capabilities starts from each of the capacity carried by each of the cells in our body. Alright. So the second one is basically the definition of cell, as it were. Alright, I think I've explained already. All living things are made up of cells. That is true. Either that living thing exists as multicellular, which means it has 2 or more cells, or it is having just one structure, which is just a cell on it. So either way, every living thing is made up of a cell. All cells comes from pre-existing cell. That is a bit long story in a way, but the summary of that is that every cell we see came from one cell before it, either by division or something, but they came. It's not just spontaneous formation. Spontaneous formation that the cell just started from nowhere. No, cells came from one cell gave us to another cell. Then the last one is multicellular organisms are made up of cells. And when we say multi means 2 or more cells made up of cell majorly. So that's what we call them multicellular. That means they have 2 or more cells. That's the basic theory of cells. Now, forms in which cells exist, they exist as a single or independent unit. They exist as filaments, as colonies, as part of a multicellular organism. Alright, so let's start with that. Now, as a single unit, the best example we can use in this case is our amoeba. However, we have plenty of those. So this is a single unit. As a single unit, this is amoeba that has lots of organelles. Just few are shown here. Now that means that we should know that amoeba can carry out all life functions. Talking about the the characteristics of living things. It can respond, it can move, it can respire, it can feed, that's nutrition and the likes. All right, so this is a cell as a single unit or independent unit. That means it can stay on its own. All right, then we have it as a filament. Now the cell as a filament talks about each cell forming a straight line. You can see it here. You can see that we have, this is a filament, this is a filament, this is a filament. So this is a spirogyria joined to this, to this, to this, to this, to this, to form a filament. What we are seeing is actually conjugation taking place. That sexual reproduction is spirogyria. So this is a spirogyria, just one. It's another one here, it's another one here. So they exist as what? A filament. So this is a cell on its own, but because they form a straight chain, we call it a filament. The other organisms too that also exhibit that, we'll get to see that in a moment when we see the differences between filament and colony. Alright, like I told you, always feel free to pause the video at any point so that you can write things down or get to hear some things properly. So another way cell exists is as a colony. Now you can see this now. This is a volvox. These are volvox forming a colony together. Now before I move on, you should bear in mind that it's not only volvox that does that. I think Pandorina also forms colony. For filaments, Oscillateria forms colony. Anabenia also forms. For spirogyria, for the filament, spirogyria is an example I just talked about. Other ones are Anabenia, Oscillateria. All right. So therefore, colony, we have bacteria can also form colony. Sponges can also form a colony. All right. So it's not just volvos that forms colony. All right. Alright, so now we look at the differences between organisms that form colony and the ones that form filament. You can pause the video here to write it down so that I can get what it is. Alright, then now we move on to what are the cells forming parts of a multicellular organism. This is a human here that has different systems, that contains different organs, and the organs contain different tissues, and different tissues contains different cells. So you can see human cells here. So looking at the brain, it has neurons in it. Looking at the blood, has different cells in it. So the cells of the brain is the neuron. The cells of the blood could be red blood cells, could be white blood cells. The cells of the skin or tissues of the body, see it here, columnar epithelial cell, that's type of cell. Muscles cells also have theirs. So it means that this old human is made up of different type of cells, like such as this. However, this is not all the cells there is in the human body. There are lots of those. For example, this is smooth muscle. The skeletal muscle was not even represented. All right. So just bear in mind that the human body or other multicellular organisms are made up of lots of cells. So these are the organs in humans, but also plants also have their own organs that apparently contains cells. So these are few organs in plants. Leaf is an organ, fruit is an organ, this is onion bulb is also an organ. All right, so these are the forms in which cell exists. Then cell is a microscopic structure, very microscopic in nature. So how do we get to see the cell? Because the ordinary eyes cannot see it, we then need to use microscope. All right, this is a microscope, and this are its parts. You can actually pause the video to see that. This is a drawing, but this next one is a picture of what a microscope looks like. There are different types of microscope. This is light microscope because it uses electricity to work. All right. However, there's another type of light microscope which I didn't add that in here, which is called electron microscope. That one uses electron, say photon of light, to see, a bit to magnify things. So light microscope only use light and enlarges the object of the, yes, majorly. Some things will not be seen in light microscope. However, if it is viewed under electron microscope, everything within that cell is being seen, because it scans through the cell and shows everything it contains. But light microscope only enlarges. However, light microscope has advantage in the sense that when you use light microscope to view an object, the object stays alive. You can still use that cell afterwards. But if electron microscope is used to view an object, I'm sorry for saying object, a cell, that cell will die afterwards. So that's the advantage of light microscope over electron. However, electron microscope sees things in a very clearer and bigger way, all right, because it shows virtually every structure within that cell. Now moving on, we have plant and animal cells. This is an animal cell which has a spherical shape. You can see the different organelles it has. Now I will quickly say some few things. This is the mitochondrion, which is the site of cellular respiration. Respiration is defined as the process whereby food substances are made or are used to release energy, and that takes place in some number of steps. All right, this is Golgi apparatus, which is the ones that packages the structures within it. You can see vesicles here. These are the vesicles, and vesicles are structures that are used to pack things within the cell. So Golgi apparatus is referred to as the packaging unit, processing unit, say distributing unit. Sometimes in some books we call it the post office of the cell, because it will distribute, to package, it will how would I put it, it will post within the cell. So the Golgi apparatus helps to post things around the cell. All right, they will now have the what's it called, endoplasmic reticulum. It has a rough side and a smooth side. So we have the smooth endoplasmic reticulum and the rough endoplasmic reticulum. The smooth endoplasmic reticulum helps to form lipids, while rough takes part in formation of proteins. And why is it rough? Because there are ribosomes attached to its surface. All right. Then moving on, we look at these are free ribosomes. These ribosomes, what's ribosome for? It is a site for what? Protein synthesis. Some books will call it the protein factory of the cell. All right. Then we look at centriole. Centriole is it contains microtubules, which is a contractile protein which is needed in what? Cell division. When you see my video under that on this segment, probably that should be part 4. So you get to see how centriole microtubules that form some spindle fibers helps in cell division. Then we look at okay, we see the cell membrane here, which forms the semi-permeable structure. All right, then we look at the nucleus, and nucleus is the brain box of the whole cell, because it controls every other activity within the cell. All right, so this is the nucleus. All right, and I will say at this juncture that a cell is divided into 3, which are, I mean a cell, I didn't say animal cell, I mean a cell generally, which is the plasma or cell membrane, the cytoplasm, which contains these organelles, then the nucleus. I take that again. A cell divided into 3: cytoplasm, sorry, I beg your pardon, cell membrane or plasma membrane, cytoplasm, and nucleus. So it's now within the cytoplasm we now have all of these. Then we have lysosome. Lysosome is an organelle that contains hydrolytic or say proteolytic enzyme that is used in digesting things within the cell. digesting things within the cell, maybe organelles that are not useful anymore. It digests them by releasing hydrolytic enzyme or proteolytic enzyme on them. Or it also digests things that enters the cell, anything that enters the cell from outside. The lysosome will release its content onto it and digest it. Now, when the lysosome digests things within the cell, that is called, things that were already in the cells before, that is called autophagy. AUTOPHAGY. While if it is digesting something that enters the cell, is called heterophagy. HETEROPHAGY. Phagy always has to do with engulfing or feeding. Alright. Now I explained something long. I said cellular respiration. Now cellular respiration, we have two types. We'll get to see that later on. Anaerobic and aerobic. Aerobic has to do with breaking down glucose in the presence of oxygen, while anaerobic is breaking down glucose in the absence of oxygen. Now, for breaking down glucose in the presence of oxygen, it takes place in three stages, which are glycolysis, citric acid cycle, which is otherwise called Krebs cycle, then electrotransport chain. Now, bear in mind that for every cell, cellular respiration takes place, whether plant cell or animal cell. All right. Now, for that, the glucose will be broken down in the cytoplasm by a process called glycolysis, and in that we have, at the end of the day, look at this here, to form this first stage, 2 ATPs are used. All right. Then to form, at the end of this day, we are able to form 4 ATPs. But because we have used 2 ATPs at the beginning, that will be 4 ATPs minus 2 ATPs we used here. So at the end of glycolysis, we have 2 ATPs formed. Alright? You can pause the video to write this sequence. It's not something hard. When I take this topic on its own, I will explain it further. Alright? So at the end of glycolysis, we have 2 ATPs formed. Now the pyruvate that is formed at the end of the day, we have 2 pyruvates formed. So each of the pyruvates will enter the what? Krebs cycle or citric acid cycle. At the end of that, so one pyruvate will give us 1 ATP. Second pyruvate will give us what? 2 ATP. So at the end of Krebs cycle, we have 2 ATPs formed. Now, what is formed there at the end of the day will move on to what? Electrotransport chain, which is a very long process. At that place, we have 34 ATPs formed. So let's do the math. 2 ATPs at glycolysis, which takes place in the cytoplasm, plus 2 ATPs formed during Krebs cycle, which takes place in the mitochondria, plus 34 ATPs formed, that forms 38 ATPs formed at the end of aerobic respiration, not anaerobic. Now, these two processes, Krebs cycle and what? Electrotransport chain, takes place in the mitochondria. So that takes us to what I said earlier on, that mitochondria is the site of what? Cellular respiration. Alright, so let me still show you that again in case you've forgotten what mitochondria looks like. This is a mitochondrion singular. Alright, so this is where Krebs cycle and electrotransport chain takes place, and this cytoplasm is where glycolysis takes place. So let's move on to plant cell as we round off. This is a cross section of a plant cell. Now this diagram is not properly drawn. This nucleus of plant cell is always towards the side, not at the center. Take note of that. And this of course this is correct, every other thing here is correct. You can see that the plant cell have chloroplast and it has cell wall, which makes it rigid. So a plant cell will always be having a rectangular shape because cell wall is rigid, and it has chloroplast, which enables it to what? Photosynthesize. And the membrane for this vacuole is called tonoplast, T-O-N-O-P-L-A-S-T. all right. Then this is centrosome, which helps the plant cell to also divide. Now let's look at the differences between plant and animal cell, which is the concluding aspect of today's class on this segment. You might have to pause the video at this instance to just write this down. This is very simple and illustrative enough by itself. So I believe you'll be able to get one or two things from that. Our next video on this series of cell will be cell and its environment. To ask me more questions, you can talk to me via these platforms. So like I said, our next video will be this. Stay tuned. Thank you. God bless you.